So can you all see the slides? Good. Okay, great. So first, I thank the principal and all others for this opportunity to present a very dear topic to my heart, which is on the adipose biology, metabolism, and energy expenditure in physiology and pathophysiology. I thank the organizers, Arulmigu Kalasalingam College of Pharmacy and the IPA South Tamil Nadu branch for sponsoring this wonderful seminar series, the virtual learning series. I'm very excited about not just I'm giving the talks, I'm excited about this opportunity. And I have been seeing the other topics that have been very well covered in this program. So it's very exciting. And I thank this opportunity. And then I would like to move forward with my talk. So today's talk would be a little bit different from what you all would have heard or would have had previously. I'm not going to talk about hardcore research data from my laboratory. The reason is that I organize my talk in such a way that I want our young scientists and future entrepreneurs in the school of in your pharmacy college to get excited about this topic and then come up with several new drug discovery ideas and also expand the adipose biology. But on the other hand, I'm also going to give some kind of a basic cell biology for adipose tissues and then talk in favor of adipose tissues being one of the targets for drugs in the future for treating metabolic diseases. So having said that, I set the lecture outline talking about health and disease, and then move on to the metabolic disease, and then set the prime for obesity, introduce adipose tissue, talk about white, brown, and beige tissue. And beige tissue is something that my laboratory has been working for quite some time now. A lot of research has been done. Beige is nothing but it's not a very big secret. It is nothing but the brown tissue in white. The white tissue gets converted into brown, and we are going to talk about some of the basic mechanisms and the so on and so forth, the transcription factors that regulate adipogenesis, which is the process by which the adipose tissues get matured into functional adipocytes, and then how the adipose tissue differentiation and transdifferentiation are regulated in the body. So this is very important to understand how a simple differentiation and transdifferentiation would set the process of obesity or to treat obesity. And then I'm going to define some of the adipokines and their functions, then move into the meat of another big matter on the peroxisome proliferator activated receptors, which are transcription factors that not only regulate the adipogenesis, but are also important for the adipose tissue differentiations and transdifferentiation mechanisms. And finally, go into the couple of slides about the current drugs that are both approved and investigational strategies that are available so far. So having set the lecture outlines, what I'm going to do is that I'm going to provide the lecture in a very simple fashion. And I would request the audience to hold on to their questions until the end of the talk so that we'll be able to have a very good discussion in a time-constrained manner as well. So health and disease. So health is a state of complete physical, mental, and social well-being. We all know that the whole world is turmoiled by the state of health right now. So we have COVID, and we are all worried about this invisible enemy. And this affects the health and causes a big disease or a pandemic. So how do you define a disease? A disease is any disturbance or an anomaly in the normal functioning of the body. A disease possibly has a specific cause, and it also provides certain identifiable and accountable symptoms. So most of the time, the etiology as well as the origin of the disease sets the symptoms that are diagnosed, and then a therapeutic measure is advocated. So in terms of disease, they are broadly classified into acute, which are rapid onset disease, and chronic disease, which are long-term. I don't want to go into petty examples here, like a fever could be an acute disease, arthritis could be a chronic long-term disease here. So all these diseases have some kind of a damage that they cause to the body. The damage could be a permanent damage or a temporary damage. And the temporary damage is reflected by acute, and permanent damage by the chronic diseases. If any one of you have a difficulty in hearing me, please let me know, okay? I can definitely crank up my examples here, like a few. So basically, the diseases are classified into infectious diseases. They are caused by pathogens. COVID is a classical example. Non-infectious disease, they are non-pathogenic. Like we have diabetes is a non-infectious disease. Metabolic diseases come under the non-infectious disease category. Genetic diseases are mainly comprised of the gene abnormalities. So today we are not going to talk about genetic diseases, although diabetes and obesities do have a base or a larger component of genetic abnormalities as well. But on the whole, I want to focus my talk towards degenerative diseases. So what are degenerative diseases? They have a progressive, or they cause a progressive deterioration of functions in the body. So they cause a slow progression. They cause maladaptations in the body. In a metabolic disease, which is a degenerative disease, there are errors in metabolism. In a neurodegenerative disease, there is a progressive neuronal cell death leading to deterioration in the functions. So in my lab, this is the only slide I'm going to put as a relevance to my lab research. So in my lab, we focus on investigating the regulatory role of a protein called transient receptor potential protein. And this superfamily of protein has 27 isoforms, and we work with 4 or 5 isoforms. And as I put it in the larger umbrella, the TRP channel proteins in metabolic and degenerative diseases, and we are focused on obesity, fatty liver disease, pain, and neurotransmission. You all might definitely come up with a question, how am I qualified to talk about obesity? This is where my lab research comes into play, and we are currently developing a drug product for obesity that induces the conversion of the energy storing white fat into brown fat. So I'm going to talk a little bit about these mechanisms, but I'm not going to talk about any hardcore data that we get from our lab. The second disease we are interested in is fatty liver disease. Third is pain. Fourth is neurotransmission. I'm not going to get into any of these compartments. I'm going to mainly talk about obesity and how adipose biology, the adipose tissue biology, culminates into a kind of an obesity a degenerative disease, and how this could be targeted for a viable treatment or therapeutics. So in metabolic disease, we know that they can be anything associated with small molecules, carbohydrates, proteins, lipids, nucleic acids, or they can be affecting certain organs, lysosomes, mitochondria, peroxisome, or endoplasmic reticulum, or they can have the whole metabolism that lead to obesity, diabetes, dyslipidemia, vascular dysfunctions, and cardiovascular disease. But today's seminar is going to be on obesity, and I'm going to focus mainly on how the obesity is produced from an adipocytic dysfunction. So we all know that metabolic diseases, especially obesity, increases the risk of all other diseases like type 2 diabetes, dyslipidemia, hypertension, so on and so forth. The prevalence of the obesity in the United States has increased. And now if I have to tell you, in the whole world, 1 out of 3 adults has one or other form of metabolic diseases. 2/3 of the world population exhibits metabolic dysfunctions, especially obesity. and medical costs associated with obesity is $250 billion. It's a pretty good market here. So this is where I get excited. We have so many young pharmacists, Pharm.D, Master of Pharmacy, and again, people who have the entrepreneurial ideas. They all can definitely have their own pitch and have a part of this $250 billion market. So obesity not only presents a disease, but also presents an opportunity in the hand of new drug development and new assay development. So what is obesity? So anything, BMI, the body mass index, 30 or greater than 30 is defined as obesity. We know that. I provided a small table below here, how we classify the BMI and then how we classify those BMI to the classes of obesity. So we think that 18.5 to 24.9 is normal weight. Now people are talking about even these numbers can have pre-obesity or pre-diabetic. We all overlap among these. So this BMI is simply calculated if we know a person's weight and if we know a person's height, then we can just divide the weight by height in meters square of the height to get the BMI. So this can be done, and I would recommend every one of us to get it done at home. And once we are in any of these numbers here, I'm talking about 24.9 and higher, we should do a lot of exercise and dieting, okay, and avoid such kind of nice donuts, okay. So anyway, what causes obesity? This is a phenomenal question. So whenever we have our energy intake and energy expenditure, there is an optimal balance between these two. So when the energy intake increases, when we take more food, this balance is disturbed. So there is an imbalance leading to excess of energy intake, and there is reduced energy expenditure that causes obesity. It's so simple. Obesity is not caused by a virus or anything. It's caused just by increased energy intake and decreased energy expenditure. So why do we worry about this increased energy intake and energy expenditure? Because everything that happens in terms of metabolic control happens in certain forms of tissues in the body. And one of the major tissue that regulates this is the adipose tissue. So what is an adipocyte or what is an adipose tissue? An adipose tissue is a specialized connective tissue, and it mainly functions, the simplest function of the adipose tissue is to store the fat in the form of triglycerides. So if you take a section of adipose tissue and view it under the microscope, what you will find is either you find a large ball of fat accumulated in white adipose tissue or several small spherical balls of fat accumulated in the brown adipose tissue. So what happens is that they all contain triglycerides, and triglyceride, as you know, is a simple combination of glycerol and free fatty acids. So the adipocytes perform two important functions. I'm going to just break the iceberg now. One function is that adipose tissues produce fat. So they process the lipogenesis. Number two, they break down fat so they can promote a process called lipolysis. So what is lipogenesis? Lipogenesis is nothing but combining the free fatty acids with the glycerol, and lipolysis is breakdown of these. So the free fatty acids that is released or that after lipolysis is usually metabolized within the mitochondria, and the process is called the beta oxidation process, the fatty acid oxidation process. And this fatty acid oxidation leads to burning up of this fat to produce heat. That's what the predominant function of brown adipose tissue. So I'm going to go through that repeatedly a couple of times. So adipose tissues, they are broadly classified into two types. A white adipose tissue, I abbreviate it as a VAT. So whenever I use the term VAT, it means white adipose tissue. And brown adipose tissue is BAT. So this BAT doesn't have any relevance to cricket. It doesn't have any relevance to the BAT. Okay. I'm talking about the bad that could cause a viral disease. So don't worry about it. So an inducible, apart from these two kind of adipose tissues, there is an inducible program that leads to the browning of fat, which means the white fat will get converted into a brown-like phenotype. And this brown-like phenotype is called the beige adipocytes. So remember this, white fat, brown fat naturally exist. And there is a pool of white fat, the VAT, that has the propensity or the capability to get converted into a beige or a brown-like phenotype. In some cases, they are called bright, B-R-I-T-E, which means brown in white. So what does the white adipose tissue does? It produces good heat insulation, good cushioning effect. We say like people, okay, chubby people, we like them because they have good adipocytes at the necessary place. Okay, so there is a mechanical cushion, which is important, and it is also provides a source of energy. So the white adipose tissue simply stores fat so that the fat is an expendable source of energy. The second class is the brown adipose tissue. And brown adipose tissue would burn this stored fat to produce heat. And the process is called a non-shivering thermogenesis. Why I call it as a non-shivering thermogenesis? Because shivering thermogenesis happens mainly in the muscles. So the muscle contraction increases, and that produces the heat for the body. Whereas brown adipose tissue is important for body thermal regulation via non-shivering thermogenesis. It has its own specific proteins in the gene paradigm that would stimulate thermogenic mechanisms within bat. And once again, I'm reinforcing that they perform main functions, lipolysis, maintain triglycerides, and this is very important. The actual level of these triglycerides as well as the free fatty acids, they will determine the insulin resistance. If triglyceride level increases, insulin resistance would occur. If free fatty acid also increases superbly without any metabolism, insulin resistance would be the result. So adipose tissues specifically function as a sink for not only fat metabolism, but also for glucose metabolism in the body. So this is a simple cartoon for us to know what would be the difference between a monolocular or a monolocular white fat and a multilocular brown fat. You see here beautifully this big white round, what you see here is one simple fat inside the big adipocyte. You see how the nucleus and the mitochondria are pushed to the sides. So this is the monolocular or unilocular white adipose tissue. You see, this is a histology picture that is given in the bottom, and the whole adipose tissue contains several amount of these big droplets too. Again, in a multilocular, you find so many smaller spherical droplets, which are generally categorized for the brown fat. And an important distinction between these two is the number of mitochondria. So you will find more number of mitochondria, more functional mitochondria in the brown fat compared to the white fat. So remember this. This is very important for the thermogenesis and thermal functions of brown adipose tissue. So against the white adipose tissue, when I say what are the subclasses of it, so there are many visceral fat, epididymal, subcutaneous fat, every fat is available in the, I don't know how this one came up. Okay. I don't know what is the reason for this one coming up, but anyway. Wait a second, let me see. Which option, sir? I don't see that on my screen, so.
Hey, Baski, on top of your screen, right,
Yeah,
there's a view option button. Just
okay.
move your icon to the top.
Yeah, there is a view.
View options, and that annotate there's a function. If you disable that, it should go off.
Zoom? Maybe I don't find it on my Mac. So is there any other? Oh, there you go. Oops. Sorry for this hiccup. I see annotate. So great. Thank you, Aditya. It's pretty good. Can you all hear me back? Good. Okay, so the function of the bat is to transfer energy from food into heat. This is the process of thermogenesis. And as I also mentioned before, BAT is essential for the classical non-shivering thermogenesis that happens in the body, which is the proper thermogenesis by food or proper thermogenesis by burning of the fat into heat. And the rate of thermogenesis centrally is controlled by hypothalamus in our brain. So that is an interplay of thermogenic functions between the brain and the adipose tissue. So this is a big a paradigm, I would say, or a controversy in the field, whether we need both hypothalamus and BAT have to function in unison for this thermogenesis. And some of the data that, especially from our lab, we think that activating independently BAT thermogenesis would be beneficial in treating obesity. So I don't want to get into any kind of those arguments, but still, for this talk's sake, we think there is a central control by hypothalamus and peripheral control by brown fat. So when brown fat is active, the lipids and the glucose are combustion. That's why I mentioned the term, the adipocytes serve as the sink for all the glucose, fat, so on and so forth. And when we talk about adipocytes, it's important for us to know these brown fat, do they exist only in animals or only in hibernating animals? No. We human exhibit or we express so much of brown fat in several of these tissues as well. We have a pure brown fat, and we also have a beigeable brown fat in several of these tissues. So we have both white fat and brown fat and beigeable white fat in our body. So now I'm going to put a simple summary slide to describe the characteristics of both white fat and brown fat. And as you see here, the white fat is unilocular. It has a large lipid droplet. It stores the fat for future energy production. It has fewer mitochondria, and it does have or doesn't have, depending upon the fat depot, a protein called UCP1, which is the mitochondrial uncoupling protein, which is also called thermogene. I'm going to talk about it in the next slide. And brown fats, on the contrary, are multilocular and have several mitochondria, and they participate in lipolysis, fatty acid oxidation, and they express so much of UCP1. And one more contrasting feature between white fat and brown fat is the presence of a transcription factor, which is called PGC1 alpha. I'm going to reflect this transcription factor once again in the later part of my lecture. And PGC1 alpha is expressed as a lower level in the white fat compared to the brown fat. I told you about the UCP1 thermogenin, or it is also called as the thermogene. Okay. The mitochondrial uncoupling UCP1 is very important for the conductance of the inner mitochondrial membrane. So when mitochondria produces ATP, it is used for producing, means utilization for energy. Now, when UCP is active, it does not let mitochondria to produce ATP. Instead of producing ATP, it uses the energy to burn into heat. That is how the brown fat thermogenesis works. So when UCP1 is overexpressed, they decrease the mitochondrial membrane potential, as a result of which the brown fat would use the energy substrate to produce heat instead of ATP. Under normal conditions, when fatty acid oxidation is decreased, more of this fat plays a role in generating more ATP and energy, and that excess energy is stored in the white fat. Whereas when you activate UCP1, which is expressed more in the brown fat, you will produce a lot of heat. This process is called thermogenesis. So when you talk about all the adipocytes, I want to talk a little bit about the progenitors of these adipose tissues. What are progenitors? These are the stellate cells or the somatic cells from which all these adipocytes originate. So all these progenitors, when you say, they have to have either a myogenic MIF-5 lineage or the absence of this lineage. So the brown fats are pure MIF-5 expressing cell lineage originators. Whereas the white fat, they predominantly originate from non-MIF-5 lineages. But there are variations here, which I will explain to you momentarily. But a subpopulation of these white fat, especially I talk about the subcutaneous white fat here as a subpopulation, or some part of the epididymal white fat, some portion, not all of them, can be derived from the MIF-5 positive lineage. So brown fat is pure MIF-5, white fat is a mixture of MIF-5 lineage and non-lineage cells. Since this heterogeneity is very important, because the MIF-5 lineage positive white cells are capable of becoming brown cells. So this is a wonderful opportunity for several drug molecules to target and induce the conversion of this white fat into a brown-like phenotype. I'm going to talk a little bit in detail very soon. Can you all see this cartoon? This was published in Nature in 2014. It's an excellent paper that talks about the MIF-5 negative and MIF-5 positive mesenchymal precursor cells. So this is what I told about these adipocyte precursors or the stellate cells. And these cells mature into either a VAT adipocyte precursor or a BAT adipocyte precursor. So the MIF-5 negative gets into this, and MIF-5 positive gets into that. Now, among these the VAT cells, they get into mature white adipocytes, which generally not express UCP1, whereas there is a subpopulation that is a beigeable, or the VAT that can get converted into brown. This is the beige fat that do express the UCP1. And then I put in a double-headed arrow here that shows that there is a transdifferentiation possible between the pure white fat to the beige fat. On the other hand, the brown fat, the BAT adipocyte precursors, would give rise to a full functional brown adipose tissue. Again, this is just brown in white, and this is total brown cells. Also, there is a separate lineage of muscle cells, these satellite cells in the muscle. I'm talking about skeletal muscle. And these skeletal muscle cells have the ability to get differentiated into brown fat directly. So this is another opportunity for drug discovery, how to stimulate the conversion of these muscle cells into brown fat for more energy expenditure. So as I told you, obesity occurs because of the imbalance between increased energy intake and energy expenditure. So if you have more brown fat or more beigeable white fat, then we'll be able to expend a lot of energy and counteract obesity. The next thing I want to talk about is that all these adipose tissue, when you look at it, they function, they have innumerable functions. And all these functions, especially I put in certain functions in capitalized ones. So the adipose tissues play a major role in regulating the physiological inflammation and immunity process, insulin sensitivity and glucose homeostasis, we talked about it a little bit earlier, angiogenesis, and more importantly, they regulate the appetite and energy balance. So I'm going to talk about this in the later part of my talk about appetite and energy balance. That is where most of the drugs are targeting in terms of anti-obesity aspect. Now I want to go back a little bit more into the transregulation or transcriptional regulation of the brown and beige adipocytes. This is important because I'm going to introduce you to some of these PPARs and PRDM16. These are the transcription factors that participate in the maturation of adipose tissue. So this is the cells in somite, which are MIF5 lineage. They commit to produce pre-adipocytes, and these pre-adipocytes differentiate into brown adipose tissue. And for the differentiation, we need three important transcription factors. And I'm going to talk about the EBF2, PPAR gamma, which we all know. PPAR gamma is an important insulin sensitizing protein, also adipocyte differentiation factor. And PRDM16 is another protein. So PRDM16 and PPAR gamma are important for the differentiation of brown pre-adipocytes into brown adipocyte. But just differentiation alone will not help. You need to have a functionally active brown fat. So you need a protein, PGC1 alpha, which I already described to you, and PGC1 alpha and a stimulus, whether a cold stimulus or a beta-3 adrenergic agonist. This would lead to a conversion of a brown adipocyte into an activated functional brown fat. If this is the case for brown fat, what about the white fat? So in white fat, we talked about two kinds of white fat. We have the inguinal white fat and the epididymal fat. So in the inguinal white fat, again, you have the functional, this is the white adipose tissue. The white adipose tissue get committed to produce a beige adipose tissue by the interaction of several of the proteins. We talked about PRDM16, PPAR gamma, and PGC1 alpha previously. And I'm going to talk about one more protein called sirtuin-1. Sirtuin-1 is called the central metabolic sensor. And sirtuin-1 is activated by drugs like TZD, which is the thiazolidine diones. Thiazolidine diones are direct PPAR gamma agonists, and those thiazolidine diones would activate this mechanism to convert the white fat into a beige adipose tissue. Whereas in the epididymal fat, we have two kinds of reactions happening. One is no MIF5 commitment. They just differentiate into a full white fat. If there is a MIF5 commitment, they become a beige adipose tissue. Are you all clear now? So all these transcription factors are important and essential for the functional regulation of beige and white adipose tissues. Now, another phenomenon that I want to discuss here is the transdifferentiation. I told you the concept of browning of white fat, and I'm also going to talk to you about the concept of whitening of brown fat. So let us take this example. We have the mesenchymal cells. These are the precursor cells, the stellate cells. They are activated by certain cues, the bone morphogenetic proteins or some of those cues that will convert into pre-adipocytes. And pre-adipocytes are of two main differentiations. When you have the CEBP alpha and other proteins, PPAR gamma, they get converted into white adipose tissue. Along with PPAR gamma and PRDM16, they get converted into brown adipose tissue. But having them matured, a functional white adipocyte can be converted into a functional brown adipocyte by PGC1 alpha. And this process is called transdifferentiation. If you inhibit PGC1 alpha, you may even turn down some of these brown fat into white fat. This is what that happens in the body in the case of obesity. So the functional expression of PGC1 alpha decreases, the functional expression of PRDM16 decreases, as a result of which more white fat production happens, and also whitening of brown fat happens, leading to more adiposity and obesity. Is that clear now? So when you look at the microscope, under the microscope, a pre-adipocyte and a mature adipocyte, what you see here is that in a pre-adipocyte, you just see the cells with all these stomach cultures, nice cells with their nucleus. And once they get differentiated, we used an oil red rose staining here to show that these adipocytes have lots of small oil droplets accumulated into them. So that is how the fat becomes synthesized as well as processed and stored in the mature adipocytes. So whenever we see obesity as a disease, obesity has a lot of mature white adipose tissue, and there is functional downregulation of brown adipose tissue as well as brown adipose tissue thermogenesis. So this is how the whole of obesity is regulated. And again, when you talk about brown adipose or white adipose tissue, all these adipose tissues are important because they secrete so many adipokines. I don't want to go into the individual adipokines because that's going to be an endless talk then for us. So I'm going to talk mainly about few of those that are important for the function of the adipose tissue. So as I told you, adipose tissue performs two important functions. Lipogenesis, it synthesizes by the esterification of triglycerides. So as you see here, glycerol and fatty acids combine, lipogenesis happens, they produce triglycerides. This is called lipogenesis, and triglycerides are broken back into fatty acids and glycerol by lipolysis. In a proper functioning adipocytes, this free fatty acid is sent to the mitochondria via transport proteins. Okay, I don't want to get into the kinds of proteins here. Once the fatty acids go into the mitochondria, because of UCP1, the fatty acid gets burnt and to produce heat, as a result of which thermogenesis happens. But if there is a dysregulation of lipolysis, then there is more triglycerides, more obesity, more insulin resistance. So this is the simplest pathology example here. So what happens when the free fatty acid levels in plasma increases? We all know that there is insulin resistance. Hepatic glucose production is suppressed, as a result of which there is more fat synthesis in the liver, and also it impairs the insulin-stimulated glucose uptake in the cells, in the muscles especially. So there is going to be impaired glucose metabolism and glucose handling. And also there is a significant increase in all these VLDL, LDL, triglyceride levels, and decrease in the HDL levels. Those all lead to the non-alcoholic fatty liver disease, and then the endothelial dysfunction, so on and so forth. So just an increase in free fatty acids, a profound increase in free fatty acids, leads to all these things. That's why whenever we take a diet, we look for the metabolizable fatty acids or free fatty acid levels in the diets as well. We talk about the cholesterol, metabolizable cholesterol, or the triglyceride levels. So those would certainly contribute for a downregulation of adipocyte functions. So how do you think the lipolysis is regulated in adipose tissue? It's very simple. First, I want to tell you that I'm going to show the cartoon for the lipolysis mechanisms, but I want to again break the ice here. There are two kinds of lipolysis. One is a basal lipolysis, one is a stimulated lipolysis. And I'm going to show the stimulated lipolysis process because this is a little bit complicated, and the basal lipolysis is going to be very simple. So whenever you activate the adrenergic receptor, specifically the beta adrenergic receptor, simply by noradrenaline or adrenaline, or you may have a specific beta-3 adrenergic receptor agonist. When those are activating, they are expressed on the nerve terminals. When these are activated, this leads to the production of adenylyl cyclase. And adenylyl cyclase is the product that enzyme that is going to produce the cyclic AMP. And cyclic AMP production leads to pKa, which is the protein kinase A activity. And protein kinase A will further phosphorylate HSL, which is nothing but hormone-sensitive lipase. This is called hormone-sensitive lipase because there are several hormones that would regulate this lipase. And also another protein called perilipin, which is expressed on the membrane of the adipose tissue. So this phosphorylation leads to lipolysis. So activating the adrenergic receptors leads to lipolysis, which involves two important proteins. One is pKa, the other one is HSL, leading to lipolysis. pKa is protein kinase A, and HSL is the hormone-sensitive lipase. And this is the function of the adrenergic receptor stimulated mechanism. So this is the catecholamine stimulated pathway. We talked about it. And once it produces, when it causes lipolysis, as it is shown here, the triglyceride is broken down into the diglycerides and then monoglycerides, and everything will releases a fatty acids. So leading to 3 fatty acids and 1 glycerol molecule. Whereas if you look at the basal metabolism, in basal metabolism, hormone-sensitive lipase is not involved, whereas you have the adipose tissue triglyceride lipase is involved here, which catalyzes the hydrolysis of the triglycerides into diacyl glycerol and 1 molecule of free fatty acids. So this is just a partial hydrolysis product. So you don't get a complete lipolysis. The basal lipolysis is only limited to the DG, means diacyl glycerol, whereas the stimulated one produces diacyl as well as monoacyl, so it produces a complete glycerol and 3 fatty acids. So you need these two to act in synchrony in physiology. But what happens in pathology, you don't have these HSL properly, so the lipolysis gets downregulated. pKa is downregulated, so lipolysis doesn't happen. So there is more triglyceride accumulation in the adipose tissue. Also, I want to tell you, the free fatty acids, I told you, UCP1 is important for this metabolism in the mitochondria and the brown fat. So if UCP1 is functionally downregulated during obesity, even when you have the adrenergic receptor activation leading to HSL phosphorylations, we won't get enough lipolysis happening. Even when lipolysis happens, there is free fatty acids. They remain as free fatty acids. They cannot be broken down to produce heat. So you find a lot of free fatty acids in the plasma transporting into the liver, leading to insulin resistance, so on and so forth. So now I'm going to talk in the later part of the talk is going to be on the adipogenic transcription factors. So there are 3 different kinds of adipogenic transcription factors. We are going to put them into the class of peroxisome proliferator activator receptors. These are the PPAR gamma, PPAR delta, and PPAR alpha. So I think most of us know that we will be able to target them, either a direct agonist for any of these or a pan-agonist, which can activate more than one PPAR alpha form. So how would these transcription factors act? Very simple. They bind to the specific DNA response elements. And once they bind to these DNA response elements, they can increase the transcription of the downstream process. For example, UCP1 is under the transcription control of PPAR alpha. Now let us talk quickly about PPAR gamma, which is a primary regulator of adipocyte differentiation. It plays a very key role in glucose and lipid homeostasis and production. And PPAR gamma agonists, this is one of the common side effects of them, is that thiazolidinediones, when they were used, they are PPAR gamma agonists, and they would also stimulate adipogenesis and more fat accumulation as a side effect. Initially, they are considered as a beautiful insulin sensitizers, and later on, they produce the toxicity of a rebound adipogenesis. There are 3 splice variants of PPAR gamma, 1, 2, and 3. And I don't want to go too much into those details, but let us talk about the transcriptional activity. Phosphorylation of the PPAR gamma 2 represses its transcriptional activity. So a post-translational modification has a profound effect on all these transcriptional factors. Summylation and ubiquitination. The summylation and ubiquitination increase the transcription activity, whereas the polyubiquitination of PPAR gamma leads to its degradation and inactivity. PPAR gamma in bat. This is where we did a lot of work, and other labs have also done some phenomenal work here. Is that PPAR gamma is important for BAT, or brown adipocyte development and function. It involves in the beiging phenomena, and the co-activation of PPAR gamma is essential. As I told you, I just showed you in the previous cartoon that you need to activate and deacetylate PPAR gamma and PRDM16. Both of these proteins should be activated and made to interact with each other so that the browning of VAT process can happen. And PPAR gamma activation in these brown adipose tissue improves insulin sensitivity. So we published a lot of papers and also others demonstrating that PPAR gamma and PRDM16 deacetylation and interaction is important for browning. PPAR delta, which is also called PPAR beta, it performs a major function in energy homeostasis. And PPAR delta is increased during the early brown and white adipogenesis. And it works along with PGC1 alpha, which is the PPAR gamma co-activator 1 alpha. It enhances the brown adipose tissue metabolism. And PPAR delta also plays a major role in the liver in stimulating the fatty acid oxidation mechanisms. And then I'm going to talk a little bit about PPAR alpha, which regulates the fatty acid oxidation. And I told you PPAR alpha is tightly regulating the transcription of UCP1. And PPAR alpha, when it is knocked out totally in mice, the mice are good to develop, but they show or they exhibit adipocyte hypertrophy. Hypertrophy means the adipocytes become big in size and there is more fat accumulation, so on and so forth. And PPAR alpha agonists are very good dyslipidemic agents. So we know that all these fibrates, fenofibrates, clofibrate, everything, all these fibrates are very good drugs of choice, which are available in the market for treating dyslipidemia. So the whole is simple summary slide. So we have the fibrates, thiazolidine diones, and fatty acids that have profound pan-activation activity in all these 3 transcription factors. And those agonists and even some of those modifiers or stimulators regulate all these functions: carbohydrate metabolism, lipid synthesis, lipoprotein expression, and peroxisome proliferation, UCP1, PRDM16 interaction. So there is a multitude of research going on in these, and we know that these receptors can be activated by specific agonists that are available in the market. And this is what I want to talk a little bit about PGC1 alpha. PGC1 alpha is a PPAR gamma co-activator 1 alpha, which is important for not only for brown fat activation, but also for the conversion of the transdifferentiation between white fat into a beigeable fat. And it has several isoforms, P4 alpha and PGC1 alpha and PGC1 beta, which are all related co-activators. Now I'm going to switch my talk a little bit towards the strategies to counteract obesity. We earlier saw, we learned about adipose tissue and their metabolism, number 1. Number 2, how adipose tissue transcription factors will regulate the conversion of a fat-laden white adipose tissue to an energy-expending brown adipose tissue. Now we're going to talk about what happens when a person is already obese, how a therapeutic measure can be advocated to either to promote weight loss, so on and so forth. Although this table is a little bit cumbersome, just I wanted to pay attention to only one thing. This is the basal overweight, obese BMI I have given. So under all these BMIs, a physical activity diet and a behavioral therapy should work in promoting weight loss. So Diet, physical activity, behavioral therapy would work in every BMI factor of obese individuals. Pharmacotherapy is very important when the BMI starts going above 30, because the person is not responding to the physical activity and behavioral adjustments, even the lifestyle adjustments. And then the bariatric surgery is the only option when the BMI exceeds 35, because that causes a morbid obese phenomena, which needs to be, the accumulated fat has to be removed, or the bariatric surgery would be to limit the food intake. So they do all these gastric bypass, etc., for limiting the food intake. So the cost associated with obesity is enormous. I don't want to put in a number for the whole world. So it's more than 100 billion each year within the United States. It's going to be close to 500 billion in the whole world, getting into the whole metabolic dysfunction as the picture. And consumers spend more than 33 billion annually on weight reduction products. This is very important. And of these weight reduction products, 90% of them are junk, because they are not going to give any long-term benefits. So physical activity, dietary modifications, changing the lifestyle are the fundamental aspects for weight loss. So what are the strategies? I talked about reducing food intake, which is going to be very difficult. That too, in this unprecedented time of COVID, people are, I think I was talking to one of my friends yesterday, and he said, before COVID, I was doing a lot of sports. And what do you do during COVID? I do a lot of cooking. So that shows that there is more kind of a food intake, because more love for food, a lot of free time. So reducing food intake, and then the blocking the nutrition or nutrient absorption. There are several classes of drugs that act in this class. Increasing thermogenesis. This is a rapidly evolving phase in terms of drug discovery. And then modulating the fat metabolism and storage. There are a couple of drugs that are targeted in this phenomena, especially when you look at the fat metabolism. We are talking about the PPAR, alpha agonists, etc., can help in this process. And modulating the central regulation of body weight. So bone morphogenetic protein 8B is a protein that affects the central thermogenic regulations in the hypothalamus, as well as in the periphery in the brown fat. So diet, exercise, and metabolism are the fundamental processes for weight loss. So what do you mean by a calorie intake and expenditure? That's very important, isn't it? I just gave you that you're going to take less food. What do you mean by less food, or what do you mean by less calories? So for every gram of fat, we have 9 calories. Alcohol, which is fundamentally, everybody loves alcohol, 7 calories per gram. Carbohydrates, 4 calories per gram. Protein, 4 calories per gram. When a person takes these things per gram, and we take approximately close to 500 to 600 grams of food in a day, or some people take it at one time. So the amount of energy that is expended is given below. At resting metabolic state, like we are all now resting, we are all sitting, and I'm sitting and talking, sitting and listening. Hopefully you're listening. That's what I guess. You're not sleeping. Good. So the resting metabolic rate is 60 to 78%. Thermogenic effect, just by eating, we spend 10% of the energy. And physical activity, basic physical activity, gives 15 to 30%. So this is the one that is very important. So basic physical activity, if you can increase and reduce the food intake, obesity will vanish. And then how do I define calorie, which is very important for pharmacists to know? The calorie is nothing but a simple measure of the heat energy. So when I say 1 kilocalorie, 1 calorie, the heat required to raise the temperature of 1 gram of water to 1 degree Celsius by 1 degree Celsius is 1 calorie. If the water is 50 degrees and you want to increase it to 51 degrees, the heat energy required will give you 1 calorie. And the heat or calorie is represented as energy. Some people represent it as the kilocalories per gram or per kilogram, and some represent it as kilojoules. Okay. Exercise and weight loss. Exercise is important for maintaining weight loss. That's important. But the problem with exercise is that once you stop exercising, there is a rebound weight gain. So you should not disrupt your exercise regimen. Also, diet is very easy to suggest, but it is very difficult to practice. Okay, but a combination of these would be effective. So exercise plus diet would lead a better effect, and limit the calorie intake. Remember the calorie calculation, increase the energy expenditure. That would lead to decreased weight gain. So when to consider pharmacotherapy? So I talked a lot about exercise and diets. For some people, it's not possible to follow. Then when they go to this edge of up to 30 BMI, then they have to think about pharmacotherapy. But usually the physicians think that it is important to combine exercise and diet. And if this combination doesn't yield benefit for at least 6 months, then it is important to go for a pharmacotherapy for obesity. So now, peroxisome proliferator activity receptor agonists, we talked about it. Are they all anti-obesity agents? The answer is no. They are not anti-obesity agents, but they are mainly used for treating some of these comorbidities associated with obesity, like PPAR gamma agonists for insulin resistance and insulin sensitivity. PPAR alpha, we talked about the fibrates, they are for dyslipidemia. And dual agonists or pan agonists, we have all of these, miraglutazar, etc. They are very useful for treating type 2 diabetes and dyslipidemia. But on the other hand, a PPAR delta agonists are used for anti-obesity agents, but there is a lot of associated risks and toxicities for these drugs. For example, PPAR gamma agonists, water retention, fluid accumulation, edema, weight gain, and more importantly, although they are initially beneficial as an insulin sensitizer, they also promote adipogenesis, leading to increased obesity. Several of these drugs are discontinued, but again, this discontinuation happens not because the carcinogenic effect that they have shown in the rodents, especially troglodyazone is a drug that shows some propensity for liver cancer. Their clinical safety made these concerns for their withdrawal from the market. Now, in the last 2 slides, I'm going to show some of the simplest drugs that are used currently for treating obesity. These are some of the drugs that are approved by US FDA for treating disease. The serotonin receptor agonists, okay, lorcaserin, this is a very good drug. Then opioid antagonists, they mainly work in terms of decreased food intake as well. Like you have this GABA inhibitor, topiramate is a very good agent that increases metabolism and decreases food intake. And a very successful drug now is the liraglutide, which is a GLP-1 analog. I didn't talk much about GLP-1. GLP-1 is glucagon-like peptide 1. It is expressed in both the peripheral system as well as in the central nervous system. In the central nervous system, GLP-1 controls the food intake. As we know that ghrelin, leptin play a role in regulating the food intake, and GLP-1 in the brain is important to suppress the food intake activity. In the sense, when we get hungry, when there is ghrelin, we get hungry, we start eating, there is a leptin-ghrelin mechanism happening, and when the fullness happens, you need GLP-1 bind to its receptor in the brain to suppress the eating so that we stop eating. What happens during obesity is that when there is decreased GLP-1 in the brain, the suppression to the eating or heating decreases. So there is no satiety, so people tend to overeat. And liraglutide is a very good drug that's available as an injection, but it has lots of toxicities and contraindications. And Saxenda is perfect, but it has to be taken under strict medical supervision. And then in this table, I've shown several of these drugs are under investigation now. Leptin analogs, the amylin analogs, GABA inhibitors, Wi-Fi receptors. These are the neuropeptides, agonists and antagonists. The Y2, Y4 receptor agonists, they regulate the food intake. They decrease the intake and increase the metabolism. So they are good anti-obesity agents. And Y5 receptor antagonists are also used as anti-obesity agents. Then GLP receptor agonists are used profoundly, and ghrelin, this is anti-ghrelin vaccine, so that the raise an interest for taking food is also suppressed in obese people, so that they take very less food, and that would lead to decrease in body weight. So I have now given, I'm now towards the verge of completing my lecture, and we talked about the adipocytes and what are its functions, how the transcriptional regulation of adipocytes happen for the white adipocytes, brown adipocytes, and how the white can be converted into a brown-like phenotype or beiging. And then we talked about how the PPARs, what kind of functions they do in transcriptionally regulating the important factors in the body, like UCP1 and PPAR alpha, and how they contribute for insulin resistance or insulin sensitivity, fatty acid oxidation, so on and so forth. And then we talked about the energy and metabolism, and how this energy metabolism helps in perfect maintenance of body weight or promote loss of body weight to maintain a healthy lifestyle. And then some of the pharmacotherapy that are currently available. And we are trying to add another new drug to this list, so that we will be able to enter into this market competition, as well as show that browning of white fat would be a good therapeutic option for treating obesity. And having this said, thank you very much for your attention, and I'll be very happy to take your questions. And
thank you.
I'm going to stop sharing.
Thank you.
Screen now.
Thank you, Professor Dr. Baskaran, sir.
Sure.
Thank you, sir. Sir, this is Dr. Venkatesh, sir.
Yes, please.
Thank you, sir. Sorry, sir, I could not join on time, sir.
It's okay, sir.
Sorry,
yeah.
Very sorry.
Yeah.
Due to some network fluctuations, sir. Thank you, sir. How are you, sir? How is everybody at home, sir?
Oh, yeah, all is good, sir. Yeah, all are good. Yeah. How are you doing?
Thank you, sir. Actually, you are belonging from Chennai, no, sir?
Yeah.
You're native of Chennai, no? Tamil Nadu.
Trichy, and I did my Bachelor of Pharmacy in Chennai, so.
Oh, very good, sir.
I did.
Very good, sir. Very good.
And I moved to Varanasi for my M.Pharm, and then I went to Austria for Ph.D.
Very good, sir. Very good.
And I would I've just opened it for questions. If the students have any questions.
Sir, we had a wonderful presentation, sir. Excellent. And any questions, don't hesitate to ask, please.
Yeah, sure.
Anu, sir?
Yes, sir.
Is there any chat box?
Yeah, they can put it in the chat. I can answer those questions. So.
Yes, sir. I have a question, sir.
Yes.
Sir, you have told about the obesity, sir.
Yeah.
Sir, what is the role of heredity in the obesity, sir? Because a lot of people are obese in hereditary nature. The whole family is obese. So during that time, what is the role of enzymes or proteins, sir?
Yeah, it's a wonderful question. So this is where it gets very iffy in terms of the hereditary models, because of the hormone-induced problems. You know, like we talked about the models of leptin, ghrelin. I'm talking about the OBOB mice or DBDB mice. Those are classical examples for the genetic aspects of the obesity. So when it becomes hereditary, this is still a hormonal imbalance leading to obesity. So the kind of drugs, okay, before I go to the pharmacotherapy, the kind of food intake and the exercise would definitely be able to help those people reduce their body weight to a certain extent. But again, we can easily do gene mapping or we can easily study a specific gene profile, and then we'll be able to identify what kind of genetic reason the person has for an obesity. I know this is an expensive process. Now, talking about the pharmacotherapies, the physicians and I strongly believe that targeting the browning of white fat, that's very important, would definitely be helpful even in case of the hereditary linked obese people. And we know that we have to do a lot of these research and showing this efficacy. And all these drugs, even liraglutide, we talked about Saxenda, they have not been tried in any of the genetic obese models. They are mainly tried in morbidly obese people. So to put it everything in a nutshell, genetic obesity, genetic linked obesity presents a challenge not only to pharmacotherapy, but also to diet and energy expenditure or lifestyle modifications. But targeting one or more of the pathways following a gene profiling would be able to certainly help. I know it's an expensive process, but several companies and insurance has started the discussions, so it should eventually percolate and get to all the public.
Thank you, sir.
Sure.
Sir, you have told that the regulation of lipolysis
Yes.
is going to be increase that insulin sensitivity as well as the reduction of fat metabolism, sir. So
Yeah.
currently, any research work is going on with regulation of lipolysis, sir?
Yes, yes. Okay, I want to just reiterate and clarify a little bit here. Lipolysis and lipogenesis happen under normal condition in physiology as well as pathophysiology. The problem with that, when there is a decreased lipolysis, the issue is with more triglycerides getting accumulated in the adipose tissue. This leads to the whitening of brown fat, and there is more adiposity and obesity. Whereas increasing lipolysis is possible through these adrenergic receptor mechanisms. I showed that pathway. You can activate the beta adrenergic receptors. So there is a class of drugs, the beta adrenergic receptor agonists, that are used mainly for triggering this lipolysis. And also subsequently, downstream, they also increase the UCP1, so the free fatty acid can be effectively burned to produce heat. So this work is done phenomenally. And there are other small issues associated with the beta-3 adrenergic receptor activation. So these needs to be addressed in terms of the safety. Otherwise, we do already have a lot of drugs in that class to regulate lipolysis.
Thank you, sir.
Sure.
Sir, in the body mass index, you have showed it is more than 25. Then we have to go for pharmacotherapy, like that you have told, sir.
Yeah.
But in case of obesity, like Indians, they are not too much weight, but there is a lot of storage of fats in their belly.
Yes.
So that also will be considered as obesity. For that, any specific treatments other than exercise?
Beautiful question, sir. So we love these BMI, but the BMI is highly compensated by not just the body weight. You know, if I can tell this as a joke. A patient went to a physician and said, my body weight is not the problem, but my height is a problem. That's why I have the high BMI. For my normal body weight, I have to be 8 feet and 6 inches tall. Can you give me a tonic so I can grow that height? So to put it in a nutshell, BMI is not the only way. The way is to measure is the waist circumference. This is what people or the patients suggest. So if the waist circumference is above certain level, then the person could be classified as obese. Again, you brought a wonderful point about the Indians, and I'm now pardon my gender specificity here, that is a that is about the apple and pear obesity. Have you heard of it? For a male, it's usually mostly an apple obesity, like we call it as beer belly. Am I correct? So people drink beer and they gain weight, and they put a lot of tummy fat. Whereas there are people who have the pear-like shape, so their bottom part is pretty heavy. So the waist circumference and the hip circumference both have to be measured, and then we have to put it in a particular calculation to define obesity there. So BMI is universally accepted, but it is not the only criteria. The physicians pay attention to the waist as well as the hip circumference to typify the apple and pear obesity. And there is benefit. People claim that the pear obesity is more in terms of benefit compared to apple, but I don't want to enter into such kind of argument. But generally, this is the consensus. So you should do both the waist, hip circumference, and the BMI. But for pharmacotherapy, the standard is that anyone who is over 30 BMI initially does exercise and dieting for 6 months. If they don't respond to exercise and dieting, then they go for pharmacotherapy. They don't start for a 25 BMI pharmacotherapy.
Thank you, sir. So generally, weight to be considered for good things, weight color.
Yes.
But according to the situation, is weight to be considered as a bad thing, sir.
You are right, sir. Absolutely, I would tell you. So white is not always good.
Oh, thank you, sir.
I like it. Thank you.
Sir, I have a question in this.
Yes.
The thing is, Indians, we are consuming more carbohydrate food, sir.
Mm-hmm.
But when you are going for weight loss, they are giving for like there are some Herbalife, some product are there. They are giving only protein, and they ask to cut down the carbohydrates. So consuming of excess proteins will have any influence in lipolysis process?
Wonderful question, sir. So I have to tell you, a dietary protein affecting lipolysis, I don't know a direct effect on this. But your dietary proteins, when they are there, your body is pushed into a kind of a gluconeogenesis for its immediate source of energy. So the stored carbohydrates and fat would just get depleted. That is what the whole idea of a proteinaceous diet, or in case of sometimes the ketogenic diets. So protein consumed in food directly having lipolysis, they may help because of the burning of basal fat, but they may not have a direct implication. That's what I think. Okay. And exactly, I do not know whether more protein would lead to more lipolysis. It's not a direct stoichiometry that is very well known.
Thank you, sir. So there is a question in the chat box regarding detoxification of body.
Okay.
No, that was the question. How to detoxify body? Like that. Yeah, it was asked by Miss Priya.
How to detoxify the body? That's
Yes.
wonderful. So there are so many ways to do that. Okay. So, like, I have to use some of the words here, like we use several of the thermogenic compounds in our traditional Indian, especially South Indian cookings. So we use licorice, we use ginger, we use the dried ginger. So those are nice thermogenic compounds that are also good detoxifiers. So detoxification, if one has to do, the best idea would be to stop eating a lot of saturated fat. That's number one. And then start consuming plenty of fluids and then healthy proteins and fruits. This would definitely detoxify. But detoxification alone, would it help in decreasing body weight? I would doubt that. Maybe there can be some cases in which detoxification may help. But detoxification will have an effect on the gastrointestinal microbes. So the microbe modification would lead to a better metabolism and energy expenditure. So for detoxification, there are so many ways you can do, but simplest way is follow our traditional herbs that we use in our food.
Thank you, sir. So you have told that anti-obesity treatment having lot of value, like it is more than 100 billion market value. So if we see a lot of drugs which are used to regulate the lipids, they have a lot of contraindication and a lot of side effects. Like bioglutamate, rosiglitazone also has been banned from the market.
Mhm.
And the other drugs also, they're having some influence on the cardiac functions, those things we have seen. So what is your opinion on pharmacotherapy of anti-obesity drugs?
Good. So the best way is that we should use the natural mechanisms that are existing in the body and tweaking them. That's why if you look at the table that I shared with you on the investigational new drugs, GLP-1 analogs, there is a lot of effort on them because GLP-1, the glucagon-like peptide 1, is a pretty good molecule that is endogenously produced. And this is peripherally, it's a wonderful insulin sensitizer. And GLP-1 receptors are expressed even on the adipose tissue, so it also improves the adipocyte functions. So GLP-1 is a very possible target. The second thing, as I told you, there are a lot of studies that are available, like blueberry extracts, and then gooseberry extracts, and then some of the studies that we published, the pure molecule of chili pepper, capsaicin, they all have the ability to convert naturally the white fat into brown-like phenotype. So those are the very good targets or possible targets in the future for the pharmacotherapy rather than inhibiting or the transcription factors or activating the transcription factors. That's why when I put these slides on transcription factors, I made it clear that they are useful for treating some of the comorbidities, but they are not going to be a direct therapy for obesity. So again, to summarize it, GLP-1 analogs are perfect molecules. BMP8B, the bone morphogenetic protein, which directly stimulates the hypothalamic thermogenesis control and back thermogenesis control, and then the browning of white adipose tissue. These are some viable mechanisms that are the natural way to treat obesity with less side effects.
Thank you, sir.
Sure.
So in the weight gaining, there is a role of stress. Some transmitter, chemotransmitter have influenced our body weight, sir.
Yes.
So any research is going on that also, sir. Like how that stress is influencing that lipolysis process.
Yes. So the simplest process, when a person is stressed, the person does three things. The person's activity decreases, number one. The person takes a kind of a pleasure by eating more food. And number three, the person psychologically downregulates the metabolic processes. So all these three are important facets in contributing to more weight gain. Also, studies have been done to show that stress, those who are under stress or depression, resort to a kind of a high-fat food eating behavior. So that also culminates into obesity. So stress has a profound factor for those who have, so this is, I wanted to, in the last slides, I just went a little bit quicker in terms of the GABA antagonists, etc. So those are very good in terms of maintaining a proper psychological balance, and they will have a profound effect in energy metabolism. So there is a lot of research that is being undertaken now in terms of reducing stress. So people practice that, and there's also publications. And we do have a good institutes in the National Institutes of Health, the alternative health. We talk about yoga and meditation to keep stress levels low. So several metabolic diseases, at least the effects could be minimized. So this is a very well known field. The other thing is, when a person is obese, that also makes the person psychologically more complex, and the person pushes and tend to have a kind of inferiority complex, so on and so forth. So that's going to be a vicious cycle in promoting more obesity.
Thank you, sir.
Sure.
And one final question, sir. That may look irrelevant.
Uh-huh.
That may look irrelevant, sir. So nowadays we have seen a lot of liposuction surgeries as a cosmetic purpose.
Mhm.
So what is your opinion on that, sir? Like there was the final option, or the pharmacotherapy will be better, sir?
Sir, it depends on the person. And if you're going to talk about a movie actress, liposuction is one of the best way for them to, because affordability is pretty high for them. And liposuction would provide a temporary benefit. But liposuction is not going to provide a long-term benefit, because there is going to be a rebound effect eventually. Unless we are ready to have a lifestyle modification following a liposuction, then it's going to produce a long-term effect. The best strategy for treating obesity is eat less, exercise more, have more physical activity, eat healthy food. If there is a plate full of maybe delicious Indian sweets and a plate full of fruits, the choice is ours to take what not. So that would decide. You talked about pharmacotherapy. As I told you, the physicians have a clear-cut approach here. They would not take a drug in their hands first, because the drugs are not safe. They do have extremely high toxicities. For example, Saxenda is significantly contraindicated in people who have type 1 diabetes, or people who have a kind of a neoplasm. So for those people, Saxenda, the GLP-1 analog, is highly contraindicated. So the physicians use these drugs only when exercise or the dietary modifications do not yield benefit. So to reiterate again, liposuction is a very good option, but it is very expensive, but tend to have a rebound weight gain. And liposuction is something that you remove the fat and you don't let the good fat do the good work for us. So when by liposuction, we even remove some of the good fat in the body, so that may have a bad repercussion at a later time. And liposuction is expensive, but it is doable. It all depends on the financial situations. So I cannot talk for or against it. So if it is for a Hollywood actress, liposuction is much easier. But when you talk about a common person, the best way is to take a good, healthy diet and do a lot of exercise.
Thank you, sir. Final question. Sir, you are doing a wonderful research.
Thank you.
And here, really, mostly in India, we are focusing on quantitative research. Normally, we are going to be prove the folklore claims, which may be true or not, those things. So, what is your advice to the future researchers? Some of them, of students, may also be researchers, to how to do quality research and how to select research areas. And that we need a guidance from you, sir.
Sure. The first thing I would tell you, I have to be a little bit of a disagreement in some part of your statement, because I did my undergraduation in Madras Medical College, and I did a wonderful project with my mentor, then Dr. V. Vijayshekaran, who was the vice principal of Madras Medical College. And I did a project with him on toxicological basis of clinical pharmacy. So the ideal research project doesn't have to be, you don't have to have a great facility to start with. So you should have a good idea. So this is the reason I took this adipose biology as a lecture today. Instead, I would have just showed the data that we got in the lab. That's not going to be a big enterprise for the students. Now the students know there are several questions that are left unanswered, so they have to prepare themselves for having a proper research question, number one. And then the number two is funding opportunities. So this is where the pharmacy, I would always say, I'm very proud to be a pharmacist and a pharmacy background person, because we now take any project into a translational niche in no time, which is going to be very difficult for people who have done pure biology, molecular biology, cell biology, or whatever the branch of science. Pharmacy gives you this upper edge in terms of taking a basic science project into an applied science. So that is where the entrepreneurship happens. So if you have a good idea, and if you are able to have a product, so now most of the pharma companies and some of the pharmacy institutions in India are looking at the repurposed drugs. I'm not asking a student to go to a natural product. That's what we always do. We cling to a natural product. We do an aqueous extract or a non-aqueous extract or an alcoholic extract and try to do it in a simple pharmacology. Instead of doing it, try to purify a compound with the existing facility, even semi-purified, and show a basic biological activity and establish proof of concept. And the third thing is that throughout the world, especially in the United States, I can definitely talk in favor of everyone who has the excellent idea and also willingness to work hard. There are beautiful places here. Once they have some kind of a lucrative idea, they could put that into a kind of a document and then contact people here for their either with a postdoc studies or even doctoral studies. Do not get clinged into the basics and use a kind of a simple pathway for your project. Even this can be a slow phase of development. Try to develop new and novel ideas, bold ideas, have concepts, stay strongly behind your concept, do as many experiments as you could do with the existing facilities. And we are here. For example, I can tell this to the faculty as well as students of Arulmigu Kalasalingam College of Pharmacy, that we in our school, in my lab, we always look for good students who have commitment to do good research. So if they come up with their own questions, if they have, they are welcome to discuss, okay? We'd be able to set up a maybe a kind of a disclosure agreement, or if they want a confidentiality disclosure agreement, we can set it up and then we can discuss those ideas. So be bold, think out of the box, don't get to your, the routine way of simplifying the project. Don't do Me Too projects, read a lot of books, develop ideas. If anyone, I watch this to Arulmigu Kalasalingam College of Pharmacy faculty and students. If you are stuck with any research article that you need, you don't have access to, do not hesitate to send it to me. So you can find me on easily on my website. You can write to me, and within 12 hours, I would be able to send that to you. So this is what the basic suggestion I would give. And I'm also willing to talk on behalf of our university, because we do a lot of memorandum of understanding between institutions abroad for faculty and student exchange programs. So we'd be able to have some projects and some funds available for doing those as well. So try to utilize those opportunities as well. First of all, don't feel shy. Next, come out, think out of the box. Do research boldly, ask questions. Okay. I hope I answered some part of your question, sir.
Thank you, sir. Thank you very much, sir.
Thank you, sir. Thank you, sir. Really, we.
Yes, sir. I'm sorry, I couldn't hear you.
Yeah. Can you hear, sir? Can you hear?
Yeah, I can hear you. Yeah.
Really very proud to have with an eminent personality, sir. I
Thank
hope this interaction will motivate the student and encourage them in active learning in forthcoming days, sir.
Yes.
On behalf of our management of Arulmigu Kalasalingam College of Pharmacy, hereby I express my wholehearted thanks to you and your management for giving such a informative, excellent, elaborated presentation and explanation, sir.
Thank you.
In future also, let's have a good collaboration for the development of our students' career, sir.
Definitely. So this is something I would like to reiterate what he said. So I thank you and the management for this opportunity, and also the sponsorship from IPA for this, the South TN Tamil Nadu branch. So the other thing I also want to little bit talk about is that if there is any good student who would like to apply for either a PhD or postdoc, they shouldn't hesitate. Okay. If positions are not available in our lab, there are going to be a lot of opportunities within the school or within the university. So people should should stop feeling shy and then start contacting us. Okay. So don't despair. If one doesn't respond, doesn't mean that the gates are closed. There are more opportunities for us to collaborate. And definitely when I visit India next time, I would definitely visit your facility, sir.
Yes, sir. Yes, sir. Yes, sir. When let me know, sir. Please let me know whenever you would like to plan to Chennai, sir.
Definitely. Yeah.
I will invite you, sir. I will invite you.
Yeah. Thank you.
Thank you. Thank you. Take care, sir. Take care of your family.
Thank
Sorry,
you.
I have taken much of your time, sir.
Not at all, sir. This is a pleasure. So uh, I don't
Thank you,
have
sir. Thank you
any
so much.
problem. Okay. Thank you so much.
Thank you.
And you all have a good day.
Take care, sir. Take care. I
Okay.
will never forget, sir. I will never forget your words.
Sure.
Thank you.
Thank you, sir. Thank you so much.
Bye, 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 bye.
Bye bye.
Thank you, sir.
Thank you very much.
you